It's my great pleasure to present Professor Cameron Atherby, who's from the University of California, San Francisco. Trained initially in pulmonary critical care at Harvard, and then moved to San Francisco around 2004. I think that's when I first met Cameron. We were both postdocs together at Dean Shepherd's lab, and following that time, we undertook residency at San Francisco and then became faculty, and now run a lab looking at matrix the lung, which is perfect for the Margaret to the work seminar, where you're going to talk about cell-mediated collagen degradation and the resolution of fibrosis. So without any further ado.
Great, thank you, Gisli. Thank you everyone for showing up and for the invitation. And I have different versions of this talk, so I can make it shorter to fit the 7 minutes I've lost. All right, so perfect. So yeah, this talk is going to be about cell-mediated collagen degradation and how it impacts the resolution of fibrosis and the severity of fibrosis. From my perspective and everything I'm going to present, I'm going to make it seem like it's all about collagen degradation. So I just say that up front because I do appreciate that you do have to have induction of matrix production to get fibrosis, even if it sounds like I'm not talking about that. So fibrosis, as we all know, is a major problem in many organs. And here we have a picture of an IPF lung. And one of the problems is that we don't have any way of reversing fibrosis. And people who get diagnosed with UIP-IPF typically have symptoms. So they have already a lot of scarring in their lung tissue. And while there are a few therapies that prevent the progression of disease, there's nothing out there that reverses the disease. So the reason that I got interested in this area was to try and say, I don't care how you got to the fibrotic state, but is there a way to get the fibrosis itself to recede once it's established? So as I already alluded to, from our perspective, if you have fibrosis, this balance between matrix production and matrix degradation is off, such that production is outpacing degradation. And if you think about it, probably every time we get any insults to our lung, like pneumonia or any other kind of injury, there will be some stimulus that will cause matrix production to go up. But most of the time, we're not going to be aware of it because given some time, the degradation pathways will kick in and there will be resolution or near normal resolution, such that, yes, maybe down the road, you have persistent infections, you'll know that you have an area that is scarred, but otherwise you're going to be unaware of it. And the best example of that would be just a scar that you develop on your skin, which at first is large and then recedes over time. And these are EMs of bleomycin-treated mouse lungs, looking at the healthy alveolar epithelium versus a fibrotic lung. And so the idea is that you have to have impaired collagen degradation to develop progressive fibrosis, or at least that's the angle that my lab would push. So just to get a sense of what the normal turnover of lung collagen content is, this is an experiment we did where we gave mice deuterated water. So this will allow us to identify the deuterium in hydroxyproline. And from the minute you start the mice on the deuterated water, any OHP that's tagged with the deuterium will be part of newly synthesized collagen. So we can get fractional collagens of tagged and untagged. And the main points of this slide is that even under normal conditions, this is a C57 black 6 mouth, the basal condition is 8 weeks old, that in a 2-week period, you're seeing about a 10 to 15% new collagen synthesis. And then this percentage will go up dramatically if you induce injury done with bleomycin. So it's clear that there's turnover, but what this doesn't tell us is what pools of collagen are being turned over. So is this just newly synthesized collagen? Is it equally distributed across all the different collagens, including basal membranes? What happens to scar tissue that's been cross-linked and has been persistent for a long time? Does that get turned over? And none of this is addressed by this study. This just tells us that there is normal turnover, which is around 10 to 15%. So as a reminder, there are both human and mouse examples of reversal of fibrosis in the lung, spontaneous resolution or resolution after treatment that prevented further matrix production. This is an old paper which shows the lung of an ARDS patient in the fibroproliferative phase. The panel on the left has obliteration of the alveolar space. And then with time, the patient gets resolution and near normal alveolar pathology. With the caveat that this patient died, so that's why they don't biopsy afterwards. And then a sort of more tractable example would be NSIP, here shown in a paper by Paul Noble, where you have a CT of somebody with NSIP. They have fibrotic disease, they have a pathology that's abnormal, they get a course of treatments, and 3 months down the road, their CT is cleared up. More obviously, this is apparent in mouse, experimental mouse models. So the bleomycin model, which I'm assuming everyone is familiar with, but will come up all over and over again. The single-dose bleomycin model, which people deride because of its lack of accuracies compared to UIP-IPF, is my favorite model because we study resolution and there's resolution of fibrosis. And so the generic single-dose bleomycin will get this peak fibrotic effect around 3 weeks, and then you'll get resolution. And if you look at different strains of mice, I'm not sure if I can get that thing to move. If you look at different strains of mice, the black 6 mice, C57, which is the most commonly studied for us, has this clear increase in fibrosis at 3 weeks that then resolves over time. It's subsequently published by many, many different groups. It is strain dependent, but there are multiple strains where resolution does occur. So then, is there any evidence of impaired degradation in disease? Okay, so let's see, it froze when I moved that kind of there. So there's actually a fairly large number of studies that looked at human samples in different diseases. This is going back 30 plus years, with the idea that there's an impaired degradative environment when you have disease. And the way these studies were done, they were either biopsies of lung fibrosis or scleroderma skin tissues where you loaded the same amount of protein and then looked ex vivo on how well it cleaved collagen, regulated collagen. And so you can see this is the IPF example, that there's less collagen-lytic activity of the lung biopsy sample. There's also a fairly large number of descriptive studies that look at expression of proteolytic enzymes and their inhibitors, showing that there's an imbalance favoring impaired degradation, both in IPF, UIP, and cirrhosis, the scleroderma, and then this disease, which is often a reaction to dilantin, dense fibrotic plaque in your gingiva called oral submucosal fibrosis, and this is associated with impaired collagen degradation. So none of this is mechanistic mouse work, but it does suggest that in human disease there is this impaired degradative environment. The other thing I point out is that there's a number of, I just picked 2 examples, but there's a number of papers out there that show pathways that the bleomycin model in mice, when you knock out the pathway or inhibit the pathway, you don't get fibrosis. So that's part of the studies of matrix production. However, in these studies, oftentimes, if you start the therapy after fibrosis is established, the fibrosis reverses. So here I'm showing these 2 examples, but this is also true for beta-6, I believe, blocking integrin, as well as LPA1. And my argument would be that if you stopped the production by giving the drug, then you give the endogenous pathways time to work. And so we claim this as proof of our concept. Degradation occurs by these 2 pathways that happen in sequence. So you have the collagen fibril. It gets cleaved by different classes of enzymes, metalloproteinases, cathepsins. Then you get these collagen fragments, which if they're left in the extracellular matrix will be pro-inflammatory, they'll recruit neutrophils, and they'll also are a setup for chronic autoimmune disease. But they can be worked on by gelatinases, or they can be taken up by cells for intracellular degradation. That's the part that I'm going to talk about the most. The EMs here are an attempt to prove that this collagen inside the fibroblasts is actually not nascent collagen protein, but protein that's been in the extracellular space and come back in, and the cross striations are sort of proving that it's fibrillar collagen. This is other people's work. So we're interested in the intracellular pathway, and so The logical question would be, is there any evidence that not taking up these fragments actually impacts? Because you may think, why, other than the autoimmune part of it, I would want to care about the fraction. So this goes back to work I did as a postdoc with Gisli, well, not with Gisli, but in the same time as Gisli. This was the first evidence of a knockout system, an integrin ligand called MFG8. And when you knocked it out and you gave the mice bleomycin, their baseline level of hydroxyproline was the same, but they had more hydroxyproline content 28 days after the bleomycin. The reason there's two graphs is just that at one point we're using heterozygous as the wild type controls, and that's picoserous red staining showing morphologically the same thing, or histochemically. And if we go back to the deuterated water experiments that I mentioned earlier, if you give these mice the deuterated water from when you give them bleomycin, you can see that they have an increase in the fractional percent of new collagen, but it's no different between wild type. So essentially, you're seeing more fibrosis, even though you start at the same point, in response to the same amount of drug, and it's not because of an exaggerated synthesis rates. We also looked at transcripts, but this is a more direct assay of the actual protein. So then I'm going to show you this visually. It's going to come up as a collagen uptake assay, and a lot of the slides that come later, and I'm just going to call it a collagen uptake assay. But essentially, what we wanted to interrogate was whether this was a defect in fragment uptake. And so what we did is take lung macrophages, incubate them with a FTSE conjugated type 1 collagen. And then here we did this visually, but later we do all this by flow, measure the internalization of these fragments. And what you can appreciate is that the wild type, the white column, takes up a lot more collagen than the knockout. And then over time, in the presence of recombinant protein, the knockout increases its collagen uptake near to the wild type. And so this was the first evidence that you can have a system where you have impaired uptake of collagen. We did some in vivo work here that I'm not showing, and you get exaggerated fibrosis, but not because of increased matrix production relative to the wild type. And then since we published that, multiple groups have shown, unfortunately not me, have shown that the recombinant protein can be therapeutic in the liver, in the lung, in the kidneys, pancreas, in reversing established fibrosis. And so some of these actually use the same or propose the same mechanism that we published, whereas others suggest that it's some interaction with TGF-beta, which is interesting because I know Gisli loves TGF-beta and I love TGF-beta. So there's an RGD sequence in MFG8. So there's another potential pathway, which is an interaction between TGF-beta, collagen uptake, and collagen matrix production, which I'll talk about a little bit later. So why would degradation be impaired in fibrotic states? So this is kind of the different areas that we've studied over the years. So the most obvious one would be that you just have so much matrix production that you can never catch up. At least in IPF, UIP, the fact that the disease is heterogeneous, the fact that there's quiescent periods and periods of active disease, to me would argue against that being a reason, though that may certainly be the reason in other pro-fibrotic states. We've been interested in whether there are cell types that are particularly important for this process of collagen degradation that are not getting to the appropriate sites once you have progressive fibrosis. People have published on how when you have collagen within a fibrotic scar, the 3D nature of it is quite different than what you would see, say, in the basement membrane or collagen type 1 around bronchial airways, and that this doesn't allow, especially with cross-linking, the appropriate sites for proteolytic cleavage or for uptake to be available. That may actually be one of the defects that's happened. And then this idea of sensing, which I'll also talk about a little bit later, which is why does a cell that's immersed in a fibrotic matrix not try to get out of that situation? So are there normal pathways that prevent that from happening, and is sensing impaired in progressive fibrosis? And finally, whether there's inherent properties of the actual scar itself that are inhibiting cells from engaging in matrix degradation. So this is sort of the background of what we've been trying to study over the last 10 years or so. So there have been a number of other pathways that have been described since, which I'll briefly touch on. The most important for this talk is this mannose receptor pathway, which is the canonical receptor-mediated endocytosis of collagen fragments. It's the only known pathway that takes collagen by receptor-mediated endocytosis. And the mice develop exaggerated fibrosis in every organ that's been looked at, despite no evidence of increased nitrogen production. So essentially, they phenocopy what we see again with GMO mice. There's 4 members of the family, but only 2 of them are important in this process. MRC2, which has got a bunch of other names than MRC1, and their expression is polarized, such that MRC2 is on mesenchymal and MRC1 is in hematopoietic cells. And these other 2, JEC205 and phospholipase A2 receptor, have all the same domains, including the fibronectin 2 domain, which binds collagen, but for whatever reason, they don't mediate this process. A couple other pathways that have been described: ApoE in the lung on macrophages, FAP, this collagenase containing transmembrane protein in fibroblasts, and then work on the dopamine D1 receptor, also in fibroblasts, regulating cathepsin-dependent degradation. So essentially, you have 3 pathways that have been described for MAX. M2 MAX perhaps are the type. If you feel like the M1, M2 classification is still relevant, then perhaps the M2 MAX carrying MRC1 are the subtype. I just say that because as single cell sequencing comes out, there's so many subtypes that probably people are not as excited about M2. And then in fibroblasts, you have FAP, MRC2, and DRD1. And then for other cell types, there's just this one paper that suggests that at least in the liver, endothelial cells through MRC1 can mediate uptake of denatured collagen. But this is really talking about collagen that's in the circulation, not collagen that's in the matrix. And one of the areas that we're exploring is whether other cell types can be induced to partake in this process, particularly the alveolar epithelium, where under fibrotic conditions, there's an induction of MRC2 transcription protein, which doesn't exist at baseline. Okay, so our major goal of late has been to really zone in on the MRC pathway and try to use it to better understand upstream and downstream regulation of collagen degradation, and to understand the relationship between collagen synthesis and collagen degradation. And this came out of work we did previously using a genomic screen in Drosophila with double-stranded RNA, and that was a relatively new technique, which was limited because it was in Drosophila and it required a bunch of steps from whatever we would find in Drosophila to then go into mammalian system. But 4 or 5 different things that we identified ended up leading to regulation of MRC2, which is not only expressed in Drosophila. So that was quite interesting to us. And so we decided to approach this now with the CRISPR technology, which has been available. And the reason I went to this slide was to confuse and impress upon you how if you look at MRC2 expression in single cell, the panel A is in mouse and panel B is in human, that you can see that MRC2 is predominantly expressed in the fibroblast population. So essentially, most of the regulation I'm going to show you is in fibroblasts and not macrophages. That being said, our CRISPR screen is done in a U937 cells, which are a malignant monocyte macrophage cell line, which express a lot of MRC2 and not a lot of MRC1. So they're kind of different as cancer cells mine, but they're very good for this screen, and I'll tell you why in a second. So this is what's now a generic CRISPR-I, CRISPR-A whole genomic screen. So we're not cutting anything out, we're just activating or inhibiting on the genomic screen. And the difference is that we're able to do a pooled screen as opposed to the Drosophila screen where it was plate-based. So here we're basically taking hundreds of millions of U937 cells. We chose U937 cells, A, because of their expression pattern, their MRC2-dependent collagen uptake, and the fact that there was a Nature Genetics screen that came up while we were working on this, which looked at uptake of a bunch of things, not collagen, but uptake of cargo using these same cells. So it gave us a way to compare our data to other people's screens and to try to get at whether anything we find is specific for the actual matrix or if it's just generalized endocytosis cargo. So the way the screen works is we take the cells, hundreds of millions of these cells, we infect them with independent CRISPR-A and CRISPR-I libraries, and then we have, I think it was 10,000 non-targeting guides as controls. We then use selection, which was, I believe, puromycin. We then add, after we've selected, we add the fluorescent collagen, and then we do sorting by flow cytometer. And I showed this in a slide, but this was 4 simultaneous flows over 10 hours with like 3 million cells. So this was for the person who did this work, it was a humongous effort. And then what we do is we have a low uptake bin and a high uptake bin. We're able to sort cells 5% on each side. And then once we've sorted cells into those bins, so it's low uptake of the fluorescent signal or high uptake of the fluorescent signal, we then extract the guides, amplify sequence, and then do the analysis. And I'm not going to show much data about it, but for anybody who is interested in these CRISPR screens, we did a second iteration of the screen where we took the 5% on each side, grew them up again, reselected with puromycin, and rescreened to enrich potentially for genes that were involved in the process. And surprisingly, the data was very similar between first and the second iteration of the screen. But you'll see in some of the data where I'll say CRISPR-I1, CRISPR-I2. 2 is always the second iteration of the screen. So then we did the screen, we did this preliminary work, which I'm not going to show you, with just single guides for MRC2 to prove to us that this might work. Once we do the screen, this is a volcano plot. So essentially, the log fold change, the direction is arbitrary, whether it's negative or positive. But essentially, what it's saying is how enriched are guides in the low bin relative to the high bin, or the high bin relative to the low bin. Each gene had 5 to 10 independent targeting guides with 300x coverage. So each gene has some 1,500 guides ultimately for it. And then the log fold change tells you how enriched they are, and the minus log R is a statistical analysis. And so the good news was that MRC2 had the greatest log fold change and the highest significance in the group. Maybe not the highest significance, but the combination of the two, which is what we would want in a screen that was designed to look at upstream and downstream regulation of MRC2. And then in the CRISPR-A screen, MRC2 now arbitrarily, because it's A activation, I put it on the positive side, but once again, it shows up as the gene that's most highly enriched in the high versus low bin. So now we're overexpressing MRC2, which we know in these cells will increase collagen uptake. It's showing up on our screen. So this gave us confidence. So there's a lot of troubleshooting, but eventually we got to the stage and felt confident that the screen would give us some biologically relevant information. And then we could also look at the correlation between significance of specific genes in the CRISPR-A and CRISPR-I screen. And there is a significant correlation. So some of these genes are both necessary and sufficient, and some of them are not when we look at the 2 different ways. So next we went to an array-based validation. So in this example, we took genes from both the A and the I screen, as well as some controls. We then made 3 individual guides for each gene. And then instead of having all the cells with all the different guides in there, each well had a very specific guide. And then we did the same analysis. And about 30% of the genes held on this. So MRC2 is the most obvious example, but TRAM2, which I'll speak about briefly, and CEL1, which I'll speak about more extensively, were others that we followed up and that validated. And so this, again, still the troubleshooting part of the screen. So then we looked at bioinformatics. So here we're looking at enrichment scores for different processes and ranking them. And what was interesting to us was that there were collagen biosynthetic processes. So there was a lot of collagens, which when you knock down the collagen gene, you had reduced collagen uptake. And it showed up both in this and if you looked at the individuals. And then we were able to then compare it to that nature genetics screen that I alluded to in the same cell type. And what you have in that first column is our CRISPR-I screen. And you can see those processes are all in the same direction. And then the CRISPR-A screen is in the opposite color, but in the same direction. And then these are the cargo that the other screen used. So IgG, complement, myelin, small beads, big beads. The big point is that they don't look the same as this. think there's some specificity for these processes. This is not just endocytosis or macropinocytosis or some version of cargo uptake that's non-specific, and that's what we're picking. So then we wanted to look at these genes that are in the collagen biosynthesis ontology group. Here you can see the CRISPR1 and 2 and INA1 and 2. And of these genes, we initially focused on TRAM2 because now we were starting to think that there's a relationship between synthesis and degradation. And TRAM2 had been previously shown to be important in fibroblast production of collagen protein. So it's an ER translocon protein. And there's this paper from quite a while ago showing that it's necessary for collagen type synthesis. And so we then went ahead and knocked down TRAM2 in fibroblasts. So we move everything into a fibroblast system, starting from stem cells. And so what you can appreciate is that in the Western blot, when you knock down TRAM2, you have less collagen type 1, which should be expected based on the other publication. But then when you look at collagen uptake, so this is what I was saying when I showed you the visual. So these are now all flow-based. But when I say collagen uptake, it's essentially that visual that I showed you, except it's measured by flow instead of MRC2. And we always knock down MRC2 as a control, and then we look at the collagen uptake by flow. And you can see that with TRAM2, you get this reduction in collagen uptake. It's not as much as MRC2, but it's quite close to MRC2. And here what we've done is looked at the effect on MRC2 transcript when you knock down TRAM2, as well as MRC2 protein. And we can see that both protein and transcript are reduced when we knock down. And whenever transcript is knocked down, protein goes down and collagen uptake goes down. So sometimes I'm showing you all three, sometimes I'm just showing you one, but we have all three for all the data that I'm showing. So what we wanted to do then was to explore this a bit by looking at the specific collagens that showed up. They were in that enrichment score data, but that were actually the ones that were knocked down, led to impaired collagen uptake. So here we've knocked down collagen 1A1, 1A2, 5A1, 5A3. These are all fibrillar collagens, and shown that when we knock these down, we get a modest, compared to MRC2, but significant reduction in collagen uptake. This is in U937 monocytes, which we can talk about whether they make collagen, if anybody wants to talk about that as the talk goes on. And here we're showing you the MRC2 mRNA transcript when we knock these down. And again, we have these transcripts in the setting of reduced collagen uptake. So for collagen, 5A3 wasn't significant. So in the fibroblasts, we did the same thing. We silenced collagen 1A1. The Western blot is showing you that MRC2 protein goes down, and pro-collagen Western blot, as would be expected, goes down. And then we have the densitometry quantification of MRC2 and pro-collagen showing reduction. And we can look at collagen uptake. And again, when you knock down collagen 1A1, we get a reduction, which is not as big as MRC2, but also significant. So then we wanted to do the opposite, which is to, if we're showing that knocking down collagens reduces collagen uptake, then we show that inducing collagen synthesis increases collagen uptake. And if so, is it through the same mechanism? So here what we've done is we've taken the U937 monocytes, we've given them TGF-beta, a disclaimer that when we're having trouble measuring collagen, use a collagen 1 antibody and load a ton. And when we're not, we use pro-collagen antibody because we're really interested in pro-collagen, because we're interested in the nascent peptide, and don't have to load as much. So this is U937 cells. So we have to do a lot of protein load, but we do see collagen 1, and we do see that it goes up in the setting of 24 hours of TGF-beta stimulation. We can look at fibroblasts and see the same thing with TGF-beta. So the first panel with fibroblasts is showing you the increase in pro-collagen as well as the increase in collagen uptake. And then it's showing you that MRC2 expression is also increased in the presence of TGF-beta. We use two independent inducers of collagen synthesis, ascorbic acid and LPA1, and again show an increase in pro-collagen peptide as well as an increase in collagen uptake. So now we're feeling more confident about this relationship because we've shown that knockdown of collagens in the screen reduces collagen uptake through MRC2. Individual knockdown reduces collagen through MRC2. And induction, though admittedly, TGF-beta, ascorbic acid, and LPA1 are going to do a lot of other things as well, but In this situation, they increase collagen protein as would be expected, and that's associated with increased MRC2 and increased collagen. Okay, so then we were looking for, so the important part here is that this is all inside the cell. And so we were looking for a sensor inside the cell that would hook this process together. It was something that would recognize new collagen protein and say, okay, there's a bunch of new collagen protein being made. We better upgrade the degradation pathway as a homeostatic loop. And we focused on cell 1L, which was in our initial screen in the CRISPR eyes as a hit. It's an ER protein that's part of the ERAD, so the protein degradation quality check, ER, not translocon, but system. And interestingly for us, it had this fibronectin 2 domain, which is important on endo 180 or MRC2 and other proteins for collagen binding. So it was in the right location to be able to sense collagen that's going through the ER potentially. And so we decided to focus on cell 1L. So the first thing we did is obtain these meths that have a floxed allele for cell 1L. And so when we give tamoxifen in vitro, or hydroxytamoxifen to be precise, we can see that collagen uptake in the knockout and the flox plus OHD is reduced, whereas it's not if we give vehicle. We can show that MRC2 protein is reduced in the setting of tamoxifen and floxed alleles, as well as cell 1L, but not in control. And we can show that cell surface MRC2 expression is reduced, which is relevant because only about 10% of MRC2 is at the cell surface, and you don't want to just assume that when we see a loss of MRC2, that it's a loss at the cell surface where it's active, and that this is a transcriptional effect when we look at transcripts. So we can also overexpress cell 1L and get the opposite phenotype. Increased MRC2 mRNA inducing cell 1L expression increases MRC2 mRNA and MRC2 protein. And then this was a big issue for reviewers, was whether this is ERAD independent. So just to remind everyone, the ERAD system is part of the endoplasmic reticulum that degrades proteins, essentially for various reasons. And so it doesn't necessarily make sense that this would be an ERAD effect, because you would expect that an ERAD effect where you lose cell 1L would lead to an increase in protein. The cell 1L is part of degradation. So we're seeing kind of the opposite. But nevertheless, really the only thing that's known about cell 1L is its role in ERAD. So what we're doing here is cell 1L works with this other protein HRD1 in the ERAD machinery. HRD1 was not a hit in the screen, and when we knock down HRD1, we do not see the effect on MRC2 or collagen uptake, suggestive that this is not ERAD dependent. And then we can sort of try to prove that if we overexpress the cDNA in the setting of knockdown of cell 1L, do we see alteration of the protein? So that doesn't directly prove it, but it's part of a package of ERAD data, suggestive that this is a function of cell 1L that's fibronectin 2 domain dependent and independent of the ERAD. This is just to show that in the fibroblasts we can see co-localization of collagen and cell 1L. And then we zone in on this fibronectin 2 domain. So we want to prove that the fibronectin 2 domain is relevant for this process. So what we've done here is an IP where we've stimulated fibroblasts with TGF-beta. We've IP'd for cell 1L, and then we've done a blot for collagen 1. And you can see that we see collagen 1 under vehicle conditions, more so under TGF-beta conditions. Of course, collagen is sticky, so whenever you do an IP, you have to be a bit cautious about it. Here we've done a different experiment where these are 293 T cells. We've CRISPR'd out cell 1L and then re-expressed it either as a wild type or as a construct that's only missing the fibronectin 2 domain. And then we've done an IP for procollagen 1, and then we blotted for cell 1L. And so only under the IP cell 1L wild type conditions do we see a band for cell 1L, not when we get rid of the fibronectin 2 domain. And then the right two are the inputs. So the effect of cell 1L on MRC2 expression is through the fibronectin 2 domain, because here, when we're looking at the meths again, when you have the cell 1L wild type, MRC2 protein is robust, but when you take out the fibronectin 2 domain, it's not. And you can see cell surface expression tracks that, and transcript also tracks that, as we would expect. And then the final thing about ERAD, I'm obsessed with ERAD, but probably for you guys, it's kind of irrelevant. But once you get looking at versions of ERAD, you want to, I'm actually only showing 2 pieces of the ERAD data, but I like this one. So here, what we're looking at is cells that have CRISPRed out, 293 cells that have CRISPRed out cell 1L, or CRISPRed out the fibronectin 2 domain. And IRE1 alpha, CD147, Saunich hedgehog, these are all proteins that get degraded by ERAD. And so this is a cyclohexamide pulse chase. And essentially, when you add the cyclohexamide under wild type conditions, you degrade the protein. ERAD's intact and not making any new proteins, so that's just normal that happens. However, in the knockout of cell 1L, you don't degrade the protein. But if you re-express cell 1L, but it's missing the fibronectin 2 domain, you continue to degrade the protein. So unlikely to be part of the ERAD protein. And then the final piece of this is looking at MYC-tagged protein, recombinant protein constructs with or without the fibronectin 2 domain, incubating them with rat-tail collagen, and then doing an IP for the MYC and then a blot for collagen. And so on that first blot, the panel on the far right is the collagen you recover when you get rid of the delta fibronectin 2 domain. And it's quite a bit less to absence, perhaps background levels as compared to the wild type. And then to the right is another example of the same thing. I'm sorry, is the input for the same thing. And then when we looked at when did this fibronectin 2 domain show up evolutionarily, it's not there in the yeast, the amoeba, or the roundworm. It starts to come when you have more complex creatures with more different collagens. So it is reasonable to consider that this is something that evolved as a regulatory mechanism and was not necessary for ERAD, which is a process that's highly conserved and exists in yeast. So our next question was, is it possible that this is a mechanism that's relevant in meaning that when you make collagen, fibrillar collagens, you increase the degradation pathway, that this breaks down in disease. So this data is, descriptive. First is just single cells showing that the population of cells that express MRC2 collagen 1A1 overlap. I don't know how well it shows up on the screen, but they overlap quite closely, and it's fibroblasts essentially. Then we looked at old mouse lung microarray data. So here we're looking at the correlation between collagen 1A1 transcript and MRC2 mRNA in whole lungs. And the point of this is that under saline or single dose bleomycin, as we discussed, a reversible model of fibrosis, the correlations between saline and bleomycin are very similar. On the other hand, if we look at IPF lung versus control, we see that the controls have a steeper slope. So they're making relatively more MRC2 relative to collagen. And this seems to have broken down in IPF. And then if we look at newer databases of single cell sequencing, we see a similar pattern, at least suggestive that this mechanism may malfunction in progressive disease, contributing in part to progressive fibrosis. Okay, so there's a couple more parts to the talk, but the conclusion for this part is that cell 1L is a rheostat that links collagen production with collagen degradation through MRC2, that perhaps this pathway is somewhat decoupled in progressive fibrotic disease. And then what we don't know at all is the actual mechanism here. I'm showing you no true mechanism. We don't know how cell 1L is regulating MRC2 transcript. It could actually be not going to say something about ERAD, but I'll shut up about ERAD. But we don't know that mechanism, so that's what we're working on now. So what we really want to know is how important is this process? By that I mean, is it rate limiting? Is it something that actually contributes to disease progression, or is it something that we see in mice and correlatively look at in human samples, but we don't really know? And this has been a difficult thing to struggle with. A couple of ways we approach this. One is looking at aging, which we know is a major risk factor for fibrosis. We've looked at mouse lung collagen content. So here, mature mice are 10 to 11 months of age. They're not old. And what's interesting is that if you look at lung hydroxyproline just without doing anything to the mice, you can see there's a lot more hydroxyproline in the mature mice. The first panel is just the lung hydroxyproline. The second panel is normalizing for lung weights, since the older mice are quite a bit bigger. So normally when we do hydroxyproline, we don't normalize for lung weight, but in this case I did. But either way, they both show the same thing. And then some picker serous red staining. And what's particularly interesting is if you look at the same time, same study essentially, at collagen 1A1 transcript, there's quite a bit less. Sure. So at least at this stage of their lives, when they're 10 months, 11 months old, they have more lung hydroxyproline, but at least at this time shot, they're not making more transcript, which again would be consistent with that there's an issue with degradation. We went and pulled out IPF data sets, pulled single cell sequencing data sets, pulled the controls so that we could get a robust number, and then looked at essentially collagen 1A1 transcripts over time in humans. And you can see the ages on the where it says identity. So once you get up into your 50s, 60s, and 70s, you get a reduction in collagen 1A1, 1A2, and 3A1, all fibrillar collagens in the lungs of humans, similar to what we see in the mouse data. And when we look at MRC2 in old mice, it's pretty robustly reduced in the whole lung life. So it's protein as well as transcript. And this is not just the lung, it's also true in the heart and the skin with the other two organs that we looked at so far. And then going back to this paper from Victor Thanikol's group, showing that old mice, 24-month-old mice, have impaired resolution in single-dose bleomycin. So the first panel is showing how much fibrosis they get initially, and I think it was 3 weeks and then 2 or 4 months later. And then the second panel is showing how much they resorb. So for them, a pretty big effect in aging mice on impaired resolution. For us at 10 months, we do see a difference at 8 weeks, but it's not as profound at all. But it is very similar to what we see with MRC2 knockout mice, 2 versus 8 weeks after bleomycin, where at 2 weeks, they have the same level of hydroxyproline. At 8 weeks, the wild type has resolved, whereas the MRC2 knockouts still have increased lung hydroxyproline. So we haven't actually proven it, but we've shown that there's impaired resolution with aging. Others have shown there's impaired resolution with aging. We've shown that there's less MRC2 with aging, and we've gone into human expression. So again, this is single cell sequencing data from other people. When we look at MRC2, SCGB1A1 is an aging marker. We can see that it peaks maybe at 36 to 45 or 50 to 65, and then it's pretty much gone at 76. Not gone, but dramatically reduced. We can isolate lung fibroblasts from humans of different ages from their lungs and measure their transcripts and show that there's reduction in transcript with aging. And then we have 2 different age groups in the right panel. And we can also look at collagen uptake of these fibroblasts to show that it's reduced with aging as well. So the last part of my talk, I'm still going to get into time, is the same thing. So that was one example where we think there's evidence that the process is rate limiting, that's age dependent decreases in collagen uptake. And then the second is a collaboration with Jordan Lasau and Hal Chapman. So it's predominantly their work that I'm showing here, which is looking at single dose bleomycin. So this is our Western blot showing that in single dose bleomycin, you get this robust induction of MRC2 protein. They were interested in BCL2 inhibition because there's a lot of interest in BCL2 inhibition. So that's why they did this. In their protocol, they inhibited BCL2. I mean, just for background, the BCL2 anti-apoptotic, keeps the fibroblasts alive and doing bad things. It's primed for death, but it's alive. And then you inhibit BCL2 and it just explodes and dies. And people have shown if you treat early or late, it can impact fibrosis, mostly from a prevention perspective. Claude's work is inhibiting from day 15 to day 21 for reasons that I was not involved in, but I got involved when she saw that 6 weeks afterwards, so sorry, the PV is baseline, BV is the vehicle treated, VN, the third one, is the BCL2 inhibitor known as NABI. And so at day 21, inhibition doesn't affect the amount of hydroxyproline. The 5 days of inhibition from day 15 to day 20. However, at day 42, you can see there's resolution in the BV group, which is your wild type, but there's continued hydroxyproline. And so essentially, BCL2 inhibition at this time frame is turning this resolving model into a non-resolving model, similar to what we see with either repeat dose bleomycin or old mice getting bleomycin. And then, of course, we got involved because there was more fibrosis and we're in the same institution and I get excited about stuff like that. So we then looked and showed that MRC2 RNA goes down in these whole mouse ones at the 42-day time points in the setting of BCL2 inhibition. And then in the precision cut lung slices, so essentially bleomycin treated, they get the BCL2 inhibition day 15 and day 20. Then you wait till day 42, then you cut the precision cut lung slices, you put them in vitro for a week or ex vivo for a week, and then do a blot for collagen. And so if you look at the wild type versus the Navi treated in that Western blot on the upper left, you can appreciate that there's all of these fragments in the Navi treated group, which are lacking, shockingly, absent in the wild type. I feel like we developed that more. You see some there, but almost two there. Is how it is. And then the MRC2 mice, we did them to see if it'd be similar because MRC2 protein is reduced. And you can see that the MRC2 mice at day 42, where we know this from paired remodeling, also have a very similar pattern, which doesn't get much increased, though somewhat by Navi. And then the bottom blot is looking at the actual lung slugs. So the supernatant presumably is where the fragments have floated off into, whereas when we look at the actual remnant lung, you can appreciate that MRC2 is reduced in the Navi in the top part of that blot, and that collagen 1 is so consistent with the idea that there's impaired resolution with reduced MRC2. And the mechanism here is not so relevant for my talk, but for completion's sake, the mechanism is thought to be that when you inhibit BCL2 at this time point, you activate a UPR response, and this is some of the UPR readouts in fibroblasts. And the general idea here is that when you do that, you essentially activate IRE1 alpha, you activate that arm of the unfolded protein stress response. That leads to RID, which is the IRE1 dependent degradation of transcripts, and that MRC2 is one of those transcripts that's targeted by this process. So that's the mechanism they proposed. For my sake, this is, I feel like, pretty good evidence that it is rate limiting. Turn a single dose bleomycin model from reversible to partly reversible, and that the MRC2 is down, and that's how it's suggested. So really what we're focused on now is trying to understand more about the relationship between uptake and production. I've already shown you some of the data, but trying to understand what the actual mechanism is, why an ER resident protein would be able to impact transcripts. I mean, the most obvious thing would be transcription factors, but that's ongoing work. We're trying to definitively prove that all the data I showed you about aging and UPR activation are actually due to MRC2. And the way to get at that is difficult, but we're using inducible MRC2 mice crossing with IRE1 alpha flox mice in specific subsets. And then we've made mice, or we've made cells that inducibly overexpress two of these pathways. So the MFGA pathway and MRC2, and we've been trying to adoptively transfer them into the lung and then track where they go and see if we can use that to impact the resolution of fibrosis. And we're doing that both in the precision cut lung slices and in the passive vivo. And then I'd just like to acknowledge most of this work was Michael Podolsky, Ting-Han Lee, Will McElroy, who were in my lab at some point, and then collaborators at UCSF in England, UC Berkeley, and then funding. And then that's the end of the talk. Thank you very much.
So thanks, Cameron. That was very thought-provoking. I've got a few questions. If anyone in the audience has questions or online, I don't know how we check. So you talk about resolution, and you show some lovely data showing that you get a failure of resolution when you manipulate the systems. But in IPF, what we see is not just a failure of resolution. I mean, you get progression. And the example I like to compare is the old mouse model, which is effectively just giving lung injury to an old mouse, which is the same as if an older person gets acute lung injury, they they don't get progressive fibrosis, but they fail to resolve as effectively as a younger person. So how are you going to model that part of it, the sort of progressive deposition of fibrosis, as opposed to the complete failure of resolution? See what I mean? It's a subtle difference.
So I guess, I mean, you're familiar with the repeat dose bleomycin, which is in theory, I don't know if anyone has experience with it. We've just started we have actually mice. The problem is, if you're really going to look at it from our perspective, you have to wait until they're old. So it's hard to tease out what's the effect of repeat dose versus old. But one thing we're doing, which is painful, and I'm not sure it's going to actually be helpful, is essentially using that model and looking at multiple time points before the mice get old to see if there's any evidence of impaired resolution that would contribute to the process. However, based on our data, the hypothesis would be that in that model, you're not going to have an effect on MRC2 because they already have impaired resolution, and we won't be able to prove it. So that's a confusing way to answer your question, but it's basically a very difficult. For me, it's very difficult because essentially the repeat dose model is the model, but then they get old. And the data on the repeat dose model, there's a 3-dose model, an 8-dose model, a 3-dose model, which is a practical one. Like the endpoint where they show a big difference is 6 months after the initial dose. It may even be 6 months after the last dose. But either way, they're already, then mice are already months old, but we already showed difference. So I think it's going to be very, very, very hard and challenging to actually model any of that accurately.
And is it all collagens, or is it specific collagens?
So we target fibrillar collagens. In the human data that I just showed a little bit of it, I showed you collagen 1A1, 1A2, 3A1. I've looked at every collagen, and it's not all collagens. Collagen 5A1 also seems to go down, but then the rest of them are all over the map. So as far as if you're asking the question of is the age-dependent decrease in collagen transcript synthesis related to fibrillar collagens? I think it is. But if you mean are all collagens relevant for this process, it depends on what pathway. Meaning that MRC2 targets multiple collagens, not just fibrillar collagens, whereas MFG8 only seems to target.
You showed your collagen degradation data primarily in Fibrosing. I know you did U937 as well, but in Fibrosing. You also mentioned that macrophages have MRC1, not so much MRC2.
Yeah.
Is MRC1 also doing that? Yeah.
So MRC1 is CD. It's the CD that everyone uses as a marker of. It's expressed CD206.
Yeah.
Sorry, I know it's 260 or 206. I always get caught up on that. So MRC1 is interesting because it's really a pan-macrophage marker, but then these sort of pro-fibrotic M2 type macrophages express more of it, and then it's used as a marker, one of the markers for that. And yet we think MRC1 would be more anti-fibrotic. And the pro-fibrotic nature of that is through signaling, sort of macrophage fibroblast interactions. So this is a long-winded way of saying that I don't know. The MRC1 mice in our hands have alternatively had an increase in baseline collagen content, no increase in baseline collagen content, protection from fibrosis, yet fibrosis. Sorry, we were talking about in vitro, technical problems in vitro. So it's been all over the place in vivo. So I don't really know in fibrosis model what's happening with MRC1. There's a lot of cancer literature that clearly shows that cancer macrophages that surround cancers express MRC1 that's important for collagen degradation. But I don't know how it fits into experimental fibrosis. We have the mice and them. It's been, currently we're trying to repeat model in them because the data's just been, we've done all, I don't even like we did it once. So.
Yeah, we did managed receptor knockouts.
Which one?
10 or 15 years ago.
Managed receptor?
Yeah.
What was your data? Because we've done it, I mean, I put it on the back burner because I got so frustrated with, because at times it looked great, at times they had like 3 months.
It's really variable and we parked it, we didn't really
I couldn't understand why it's so variable because it's not like in those experiments, controls were good. So I have no idea about MRC1. I mean, it's unfortunate because I'm super interested in why 2 molecules that basically have the same structure have different relatively some overlapping functions, but some very independent functions. Some of it's like the cytoplasmic tail of MRC1 has some activities that's different. But yeah, I don't know where MRC1 fits into the picture at all. And we've also had a really hard time making, we've been trying to make conditional knockouts. We were successful at MRC2, but for whatever reason, targeting MRC1 has been, even though there's no phenotype of the global that I'm confident with, because of this role, and this, I wanted to at least be able to get cell type specific, and we've just never been able to successfully target. Generally, yeah, there's some company in China that has it. It's not quality control. So, I mean, they said there's a quality control. I mean, they haven't checked if it works. They just will sell it to you. Yes. Which they said if it didn't work, they would then take it back and work on it. But you know, we're running out of money, so they're using us to sponsor.
I've got a question on the chat there. At the generalizability of the phenomenon. Would you expect to see similar effects in other age-related models with the fibrosis?
I think so. Yeah, I do think so because liver. So just read that question. So do you think? I think it's a general fibrotic phenomenon, meaning that collagen degradation is important in every organ system, especially because both the MFG8 and the MRC2 mice have exaggerated fibrosis in multiple organ systems. But you ask a very good question, which is the reversible model of liver fibrosis. We've done CCL4. What I can say is that certainly the addition of recombinant MFG8 to the CCL4 and the biliary ligation model reverses the fibrosis faster than if you don't give it. So I don't think it has to be permanent. I think it can accentuate it. And I think it's generalizable, and I don't think it's lung specific. In fact, usually we do both lung and liver when we're going with the. So in my hands, this is one of those frustrating things because I was thought doing a revision on a big time journal, and I couldn't get recombinant protein to reverse liver or lung fibrosis, but many people have published it since. I mean, many people, like independent groups. So it's kind of, I don't know what's doing that. Our PI would say they're all wrong, our former PI, but you know, interesting. Everyone else is wrong if it's you can't get to it. So I'm more equivocal. So
yeah, and you had the chance to look in out of even human data. Where is the MRC2 produced? Is it peribronchial? Is it more across all type of fibroblasts?
oh, MRC2.
Yeah.
We have a lot of single. It's very broadly expressed.
Yeah.
So we've queried other people's single cell, both human and lung fibroblasts, and it's in every subset. And I would have liked to see, you know, what would have been perfect is if like Dean Shepard has this Nature Com paper, the CTH-CR1. It would have been really nice is if those guys that make the most would have had the most MRC2. I didn't show you that data slide because it's negative, but at least that, I mean, that's a rough way to look at it, but that you know, it's not it. They make plenty of MRC2, but so do the alveolar ones. So it's pretty diffusely spread, and you know, there's a tiny bit of expression on alveolar macrophages, but it's very hard by protein, by transcript.
Yeah, but my next question was the transcript maybe largely expressed, but is a protein actually there?
Yeah. In Max, it's really just a transcript. But with those mice, we worked with conditional mice. We've bred our conditional mice into the Dean's made 5 pre-lines for pro-fibrotic, pro-inflammatory, alveolar, basal, not basal, peribronchial. So we are trying to see which of those fibroblast subsets is the one responsible for the phenotype of the global knockout, but you know, it's certainly possible that we won't see maybe all of them. Yeah. It's hard to. It'd be really nice if it was a specific. It'd be really nice for me in my publications if it was a big subset. But you know, we're doing the experiments because they're logical and we have access to them. But my guess is it's not going to be.
They could be
unless it could be none of them. And we also have a PGF alpha, so pseudo global fibroblasts. I suppose if that doesn't work, seems to be.
Do you think it could be such a conserved mechanism that there will be some redundancy between fibroblasts, just if one is missing, like one will compensate?
I'm going to guess that all of them have the capability. I think purely scientifically, it'd be much more fun if it was not considered across different subpopulations, but that's like anything else. So we're going to learn more and more about these subpopulations as our techniques get. So I'm a very negative person as far as science goes. So my assumption is that everything's going to work out in the most uninteresting, negative possible way. So that's why I think it's, and even when, when people are making all these single cell knockouts, shocked when there's a difference. But there is. So,
yeah. I think the functional downstream consequences of the change in degradation pathway. So if your fibroblast is having to munch up more collagen because it's producing more, do you think that has a knock-on effect? Do you think that some way promotes fibrosis through a sort of positive feedback loop?
So the loops that we're interested in, I'm interested in all the loops, but the loops that I thought about and tried to create, one loop that is like what we showed here. So that's one loop. The other loop is that when you take up a fragment of collagen, does that inhibit or activate further production? And you could imagine that, under normal conditions, maybe the fibroblast is saying, hey, I'm taking up a lot of collagen, so I need to make more. And then in disease, that breaks down. So that's one way to think about it. I haven't been able to prove that, but there is this interesting relationship between TGF-beta, MFG8, and collagen uptake, where what people are publishing is that, or at least people who push that mechanism, I'm going to pull off one of those papers, so I shouldn't make it sound like people strictly, but that there's this competitive inhibition between the RGD domains. So you prevent TGF-beta induced matrix production. However, they show a time scale left like 3 days from resolution. It's hard to imagine that if you shut down, once you have established fibrosis, I think the paper I'm thinking of the CCL4, they show that you establish CCL4 fibrosis, then you give recombinant MFG8, and in 3 days, the fibrosis reverses. And they say it's a competitive inhibition of TGF-beta signal. And for me, that time frame is way too close to make any sense. However, it would make sense to me that uptake of that fragment would downregulate further matrix production. And we're trying to show that. Even in the MRC2, other people's MRC2 data, it's not entirely clear to me. Like when we showed our first MFJ paper, we showed by a synthesis of protein and transcripts that there was no difference between injured wild type and injured knockout. But if you dig into the work with MRC2, it's not always shown. So there's sort of an assumption that because there's impaired degradation and there's more fibrosis, that's the mechanism. I think there's a lot of room there. And then I can't imagine that the system doesn't communicate with each other. I mean, one other thing that I didn't put in this talk is that we tried inducibly expressing MRC2 on the epithelium, and it's shown that when we do that, I'll do it epithelium. So we have a mouse, the inducible mouse. And when we do that, we can promote epithelium to get involved in the process. But we haven't yet shown that leads to a hard outcome of altered fibrosis. We just haven't done experiments.
One of the things in this system which always puzzled me is that if we look at neoepitopes, for example, collagen neoepitopes, and we did this in the profile study, the most dramatic upregulation is in collagen degradation. So, and
you mean after glio or after?
No, in patients. In patients with IPF, we get progressive disease. Ones with more progressive disease are the ones with highest rates of collagen 3, 6, 5, 1 degradation. And we always just assumed that was because this is degradation by MMP7. So that there's more collagen degradation going on because there's more collagen. So we have a lot more collagen, we can have more. And it wasn't directly linked to the synthesis degradation pathway. I remember we did analysis of neoepitopes for synthesis and degradation. They were largely flat. The ratio was stable. Didn't matter whether it was progressive or non-progressive. So we've always been rather puzzled mechanistically, why high rates of degradation would be associated with progressive fibrosis, i.e. more deposition.
So just so I have it in my mind, when you look at IPF progressors versus non-progressors, the progressors, even though they're not necessarily making more synthesis of neoepitopes, they have more degradation.
So they have more synthesis, but they have more degradation.
That's okay. I mean,
But
that's sort of what
they're
we're
not-
pushing, but it's not in proportion to each other.
Yeah, so
Yeah.
you wouldn't see that.
Yeah.
We don't see any major difference between the synthesis and degradation ratio between progressive and non-progressive.
But maybe they were more interested in us than we said.
And,
About those complexities. No, I don't know. The other thing that's I think really interesting is in that vein is that and I think we've talked about this before, so forgive me if I'm repeating, but most of the collagen, pro-collagen, doesn't ever leave the cell. Most of it gets degraded inside the cell, which, you know, when you increase in synthesis rates is a big setup for activation of UPR, blah, 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 blah. But you could imagine if normally you're degrading 70–80% of your normal collagen, if you then have a profibrotic stimulus that makes you 5x of that, then in addition to what you're bringing in, you're going to have to be degrading a bunch of nascent collagen peptide by numbers a lot more. And I've wanted to explore how that fits into the whole picture, and I haven't. But I think that might be an area to try to understand that. I think a lot of what's going on in the ER is probably it's not a novel thought.
do you think other cells are then recruited? So you could force the other cells to take up more volume. Do you think in pathological states, this desperate attempt to get rid
Well,
of
so yeah, so that, you know, I didn't show the data here, but in both the polio, single dose polio, and in IPF single cell sequencing, you see like if 10% of cells, 1% of cells are expressing MRC2 under control conditions, it goes up to about 10 to 15% in IPF conditions. So I'd like to think that that's a response and attempt to get involved. But, you know, unfortunately or reality-wise, like the MMPs, there's kind of more and more roles that are being ascribed to MRC2 that are independent of collagen uptake. They take up lectins, surfactant proteins. And so we're doing the experiments with a knockout inducible in alveolar epithelium. So we'll know one way or the other. But I could see it going in either direction. It could actually be a pathological mechanism because I almost don't dare use the word EMT because I know it's really bad. But there's some newer EMT stuff that suggests that the inducible expression, they don't say it, but if you put other people's work together, that perhaps the MRC2 is showing up in the epithelium in disease and causing EMT, and that's making things worse. So we have the tools at least to figure out what the response is to this. But I think it's equally likely that it's nothing to do with collagen uptake, but maybe it's pathogenic through a mechanism that's unrelated to. But interestingly, that the cancer studies that I put together, it ends up going back to the ER and RID in the opposite direction than what we're showing for cell 1.
If there are no further questions, I'd just like to thank you once again for an excellent.
Thank you.